disciplines that belong to environmental and applied microbiology. Okay, so initially I will be talking about environmental surveillance regarding the water and air microbiology, and later on I will be talking about milk and dairy microbiology. Okay, so when we see the indications of environmental surveillance, as such, routine environmental surveillance is not recommended by CDC, but it is generally done in special conditions. So in the hospital settings or in the healthcare settings, we generally do it when there is a we have to do any outbreak investigation. Okay, so if there is any outbreak in the hospital and we want to know the source of pathogenic organism, then or we want to know the causative organism, its reservoir, how the outbreak has occurred, then we can do the environmental surveillance. Secondly, when we have implemented any infection control practice which is new to that hospital and we want to evaluate and want to assess the effectiveness of that practice in the hospital, then again we do the environmental surveillance. Then third is when there is a construction in the hospital, so we want that the environment should be clean, or we want to commission any new building or any new room that has been constructed recently to the hospital. So we want to check whether the environment is clean or not. Then again we do the environmental surveillance. Okay, then after that we can do it for the research purpose when we want to do certain research, or we want to estimate the healthcare associated infection rates in the hospital. Then again the environmental surveillance is indicated. In the community settings, when we talk about community as a whole, so then we do the environmental surveillance when we want to know the high risk areas for certain diseases which can spread through the various sources in the environment. Secondly, again, whenever there is an outbreak investigation in the community. And the thirdly, in the food industry, because we want the environment in the food processing industry to be clean. So environmental surveillance can be done there. So routine or periodic environmental surveillance as such is not recommended, but it is generally done in certain specified conditions. So coming to the water bacteriology, when we talk about microbiology of water, then there are certain bacteria which are natural water bacteria which are present there. For example, Micrococcus, Serratia, Flavobacterium, Pseudomonas, Alcaligens, and Acinetobacter. So these are basically Serratia, Flavobacterium, Pseudomonas, Alcaligens, Acineto. They all are gram-negative bacteria, okay, which can be found as saprophyte in the environment. So they can be present in water, in the natural water, along with the Micrococcus, which can also be seen in environment. Micrococcus is a gram-positive cocci. Then there are certain soil bacteria which can be washed into water. For example, Enterobacter aerogens and Enterobacter cloacae. These two bacteria, they belong to family Enterobacteriaceae. And then there is another spore forming gram-positive bacilli, that is Bacillus subtilis. Okay. Then there are sewage bacteria such as coliform, streptococci, clostridium, micrococci, proteus. All these bacteria can be seen when they are can be seen in sewage, as well as intestinal bacteria, Escherichia coli, Enterococcus faecalis, Clostridium perfringens, Salmonella typhi. So they all are intestinal bacteria. So can any one of you tell me what is the significance of testing water quality? Can anyone tell me what is the importance of water bacteriology? Am I audible? Yes, to prevent waterborne disease. Very good, Ansh. So, okay, to check its safety, Mahika, just a sec. Okay, basically, yes, to check its safety and to know and prevent the waterborne diseases, right? So, there are certain waterborne pathogens that you must be knowing because you have been taught about these organisms. So there are bacteria like Vibrio cholerae, Escherichia coli, Salmonella, Shigella species, Yersinia enterocolitica, Campylobacter species. All these are the waterborne bacteria. Then there are certain viruses which can spread through fecal-oral route, such as hepatitis A and E. Then diarrheogenic viruses which can cause gastroenteritis in humans, such as rotavirus and norovirus. And then enteroviruses, especially the poliovirus, right? Then there are parasites, you know them, entamoeba, basically protozoa and helminths are there, which can spread through water, contaminated drinking water. So all this you must be knowing. So this is basically a broad list about the various waterborne pathogens. So now when it comes to the water testing, it is generally not feasible and practical to test all the pathogens individually whenever the sample uh, is being tested for any pathogen. Okay, so it is not generally practical because most of the bacteria which are pathogenic, such as Salmonella, Shigella, they are generally present in very minute quantity in water. So generally they will not be isolated, and also they are not very much resistant in the aquatic environment, so they die quickly. Okay, so the isolation rate is very low. So what we do is also sometimes the water is chlorinated, so most of the organisms they die due to chlorination, such as viruses, right? And so there are various factors due to which the all the pathogens cannot be tested for the pollution in the water. Okay, so what we do, we say ki we test for certain indicator organism, which basically indicates that there has been a fecal pollution of the water, because our main concern is the pathogens which are present in stool, okay, which are pathogenic to humans and which are spreading through fecal-oral route. So any organism which indicates that there has been a fecal pollution of water, we test those organisms. So what we do, we test those organisms which are generally commensal in the human intestine, and so they are present in large quantity. So they are present in large quantity in water, so it is easier for us to isolate them, and also they can survive for some time in the harsh aquatic environment. Okay, so there is a list of indicator organism that has been given in your textbooks. So generally we test for the coliforms. Coliforms bacteria such as Escherichia coli, Klebsiella, Enterobacter. But the most sensitive indicator is the Escherichia coli, that is fecal Escherichia coli. Okay, it is also a thermotolerant. It can survive at 44 degrees Celsius. So it is fecal Escherichia coli, which basically confirms the recent fecal contamination of water, and it is most sensitive indicator. Recent because after some time it will die. So if it is present, if it is being isolated from the water sample, that means the contamination has been reset. Okay, but other coliforms such as Klebsiella, Enterobacter, they can be present in soil, or they can be present as a saprophyte in the environment, and also they are able to survive for longer duration as compared to Escherichia coli. Okay, so if they are present, that then it means that it is a presumptive pollution or contamination, and also it might not be recent. Okay, so it is a remote contamination. So if there is a recent fecal contamination of water, and then the fecal Escherichia coli, which is a thermotolerant Escherichia coli, it should be present, and it is the most sensitive indicator. Then there are other organisms such as fecal streptococci, which also confirms the remote fecal pollution. It is also resistant, and it can survive for longer duration. Similarly, spores of Clostridium perfringens, they also denote remote contamination. Then Pseudomonas aeruginosa, it is a less reliable indicator because, as I told you, it might be present as a saprophyte in the environment, and it might be present in the natural water as well. Okay. Then there are certain bacteriophages which are specific for E. coli. They also indicate fecal pollution, and they also indicate indirectly viral pollution, because in the similar kind of environment, other viruses might be also present if there is a contamination. Okay, so this is the list of indicator organism of fecal pollution. So what are the different tests that we performed in microbiology for the testing of water? Generally, we can divide them into three main tests. One is the multiple tube method, second is membrane filtration method, and third is presence or absence method. So multiple tube method is extensively used for the drinking water analysis. Okay, and it can be used for highly turbid samples. So its advantages are that it can be used for highly turbid sample, and it is generally extensively used in labs for drinking water analysis. Okay. Then second method is membrane filtration method, in which these method can be used in the hospital settings when we want to test the dialysis water. You must be knowing what is dialysis. So in hospitals there are certain dialysis unit in which there is a dialysis treatment plant in which the water is being used for this purpose. So this dialysis water is basically, uh, in the end, when we are performing dialysis, it comes in contact with directly with the blood of the patient. Okay, so there is a dialysis membrane. So if the dialysis water is not pure, it is not free from microorganism, then there might be bloodstream infections in the patient who are receiving dialysis. So testing of dialysis water is must. And as I told you, in the environmental surveillance, it is generally not recommended, but this is exception in which the testing of dialysis water is periodically done. Okay, so it is generally the preferred method is membrane filtration method. Now, membrane filtration method can also be used when the bacterial count in water is expected to be low, and also it can be used when we want to test the large volume of water. This I will tell you later how we do it, but we can test large volume of water with the help of membrane filtration method. And the third is presence absence method, in which we are just seeing whether the particular organism is present or not. So various commercial kits are available for that, in which we can detect the presence or absence of a particular organism. Okay, so how we first step is collection and transport of water sample, which is important, because if the collection and the transport is not proper, then the right result will not come. Okay, so first of all, when we are collecting sample, it should be in the sterile container, and the volume is generally around 150 to 200 ml of water. And we also add a neutralizer, which is also known as sodium thiosulfate. It basically neutralizes the residual chlorine. Sometimes in drinking water or swimming pool water, the chlorine is added. Okay, so to neutralize that chlorine, sodium thiosulfate is added so that it does not interfere with the culture results. Okay. Now, what are the sampling points and location? So if you are collecting samples from the community, so like if you are testing for drinking water, then tap water, household storage container, water tanks, hand pumps like groundwater, wells, then stagnant water in the form of ponds and streams, running streams, sample can be collected, and also the water from swimming pools is also being tested. Okay, so these are basically generally different locations from where the samples can be collected and is being tested. So in our lab, we get samples from the water sample from various hostels, various uh, locations in the hospital, such as dialysis unit, as well as the swimming pool from the in the college. So we are getting samples for the testing, from all these locations. Okay, so in the hospital, basically pipelines or endoscopy rinse water or dialysis water or sinks, from these location we can collect the sample. Okay, generally those sources are taken which are basically which are generally the main sources. Okay, which generally tells that the which represents the other sources as well. So it has to be decided by the hospital infection control team about the sources or the sampling points. So these are the various points and location from which the sample can be taken. So how we collect the sample, I'll just show you the quick video. So here you can see in the first uh, point, we are taking sample from the tap water. So whenever we are collecting the sample, first we have to clean the source. So we clean the tap with the alcohol and we collect the sample in the sterile container, and here the lid of the sterile container, it should be directed downward. And here you can see that the base, the container has been hold from the base, and now the sample has been collected and it should not be filled up to brim. Okay, so first you let the tap water run for few minutes and then you collect the sample and then you label it. You label it with the you can mention the tap number or the date. Okay, and then you send it to the lab. Now, the transport of the lab should be as soon as possible, preferably within 2 minutes. But if you are using ice packs, then it can be delayed up to 6 hours and maximum up to 24 hours. Now, this is the commercial kit that they showed. It is basically done for chemical testing, such as pH, turbidity, and chlorine, that has to be done on site. Telling how to collect the sample from the hand pumps. Okay, so first you let the hand pump operate for few minutes, let the water flow, and then you sterilize it again with the alcohol swab, and then you clean it. You can also use the blue burner to sterilize it. Okay, then after that, you again let water flow, and then you collect the sample. Okay, now collecting container can be a glass container or plastic container, but it should be sterile. Okay, we prefer glass container. Then what we do, if we collect, we have to collect sample from the well. So we take a container which has to be sterile and we tie it with a stone. So we basically, we apply a weight. So we are tying it with the help of stone. And then to that, we also tie a long string. Okay, so here you can see, this is a string that has been tied to the bottle, and there is a stone also that has been tied. So now you will open the lid and you will, with the help of the string, you will let the stone and container reach the depth of the well, and you will make sure that it does not touch the walls of the well, and then you will fill it and you will pull it. The mouth of the container should be upwards, in the upwards direction, and again, if it should not be filled up to brim. Okay, so this is how we collect the sample from the well. Fourthly, when we have to collect sample from the watercourse or reservoir, what we do, we take the container, sterile container, and we immerse it within with a depth of up to the depth of 30 centimeter. Okay, now again you have to hold it from the base, and the mouth of the container should be upwards, and if there is a active current, then it should be facing towards the direction of the current. Okay, so this is how we collect the sample from the reservoir or running streams. Okay, So when it comes to the transport, we should have certain transport boxes which should be filled with ice pack. So cold chain should be maintained whenever we are transporting the sample. If the cold chain is not there, ice packs are not there, then sample should be processed immediately within 2 hours. So the first is the multiple tube method. So what we do in multiple tube method, it is also known as presumptive coliform count, because we are making a presumptive diagnosis that the coliform is present. Okay, we are not confirming the presence of E. coli in this tube method. So what we do, we have this Mcqs purple broth, which is a liquid medium. Okay, so we prepare this medium. We have this bottle, which is 50 ml bottle. Then it is a double strength. Okay, it means that the concentration is generally 80 grams per liter of the medium. Okay, so we prepare 50 ml and we dispense it in the medical flat bottle. Okay, so this is the 50 ml of double strength of Mcqs purple broth. Then we also dispense this double strength in test tubes in the quantity of 10 ml. Then we dilute this and we make it single strength. उसमें 80 ग्राम था, इसमें 40 ग्राम per liter होएगा. And we also dispense it in 10 ml. So we have different tubes of media with different volume and different concentration. So we have 50 ml tube, we have 10 ml tube, both are double strength, and we have 10 ml tube which is single strength. So ये तो हो गया culture media. Second है, now why we call it multiple tube method, because we are using multiple tubes which have specific volume of media as well as the water sample. Okay, and we are testing it. So ये table जो है, it is recommended by WHO. They have given this for treated drinking water and the water which is polluted. Okay, so in this table, these are the sample volume of water. Okay, so if suppose any water which has been treated and we want to test whether the treatment was effective or not, so what we do, we are basically using these two sets of volume. One is 50 ml of water and another is 10 ml of water. So 5 tubes, this is the number of tubes. So 5 tubes of 10 ml of water and 1 tube of 50 ml of water. Okay, and if it is partially treated or polluted water, then we are using 3 sets of volume. 5 tubes containing 10 ml of water, 5 tubes of 1 ml of water, and 5 tubes of 0.1 ml of water. So multiple tubes are being used. That's why it is also known as multiple tube method. So how we do it? Suppose we are testing treated sample. So we have taken this media. What we will do? We have taken this 50 ml of double strength media and we have taken 5 tubes of 10 ml of double strength medium. Okay. Now we will add 50 ml of water in the first bottle and 10 ml of water in these 5 tubes. Okay, so each tube will have 10 ml of water. Then we will incubate this tube up to 24 hours and then 48 hours, and then we will see the color change. Okay, so this is Mcqs purple broth, which has indicator bromocresol purple. So if the coliform is present, the lactose will be fermented and acid will be produced. So the media, the color of media will turn yellow. Okay, and here you can also see that these tubes have Durham's tube. Okay, so with the help of these Durham's tube, we can also detect the presence of gas. So after incubation, if there is a change in color and there is a formation of acid and gas in the tube, that means the tube is giving positive reaction. Okay, so now we have these Mcgrady statistical table which are available. Okay, so here we will see that if this first tube is positive, then the number one of 50 ml, the number will be 1. Okay, if it is negative, then the number will be 0. Then in the second sets of tube, if it is 0, then 5 of 10 ml the reading will be 0. Okay, so like this way, with the help of Mcgrady statistical table, we see the reading and we calculate the most probable number per 100 ml of water. So basically we are detecting the number of bacteria which are present in 100 ml of water. So these tables are basically present. Okay. So here you can see for untreated water, we are using 3 sets of tubes. Okay. So in the first set of tube, we will add 10 ml of water, then 1 ml of water, and then 0.1 ml of water. So in the first set, we are using double strength medium, and in the next tubes, 2 sets, we are using single strength medium. So in this, we will see the growth, and we will see the number of tubes which are giving positive reaction. So in the first set, we will see how many tubes are giving positive reaction, and accordingly, we will take the reading, and we will detect the most probable number which are present per 100 ml. Okay. For example, if it is treated water, then in the first tube, here you can see, right, the color has been changed and the air bubble, so gas has also been formed. So the reading in the first tube is 1. So here we can see the 1 series, and in the second set, the reading is 2. Okay. So here from the table, we will see 1 and 2. So it corresponds to the MPN of 6. Okay. So 6 coliform per 100 ml of water are present. So this is how we take reading from the multiple tube method. Secondly, so in this, can you tell me what will be the reading for this? In the first tube, first set, there are 2 tubes which are giving positive reaction. In the second set, again, there are 2 tubes. And in the last one, again, there are no tubes which are giving any reaction. So, just a sec. Okay, the answer is 9. Justice. Yes. Okay, the answer is 9. So this is how we take reading from the multiple tube method. Okay. Now we have taken this reading. We have to see what is the interpretation. Okay. So again, this is being given. So if the quality, if the reading is more than 10 or equal to 10, then the water is unsatisfactory. If it is 4 to 9, then it is intermediate. If it is 1 to 3, then it is satisfactory, and it is 0, then it is the excellent. Okay, so this is how we see whether the water is satisfactory to use, or it can be used for the drinking purposes, it is excellent, or it is unsatisfactory. Now, if the result of the tube is positive, then we do not proceed further. Okay. But if the result is positive, then we confirm it. We confirm it with the help of differential coliform count. Okay, it is generally done to confirm that the coliform bacilli detected in the presumptive test are fecal E. coli. So what we do, we subculture those positive tubes on other media, which is lactose containing medium, such as brilliant green bile broth. Okay, and then we basically incubate it in the water bath at 44 degrees Celsius. Okay, and then we see whether there is a detection of there is fermentation with the production of acid or gas or not, because it is a thermotolerant E. coli. We also can demonstrate positive indole test at 44 degrees. So if the bacteria survive after 44 degree, it will give a positive indole test. So this is also known as Eichmann test, based on the person. He was a Dutch physician who basically worked on the water testing methods. So the next method is the membrane filtration method. It is one of the method which is basically done, as I told you, mainly in the hospital setting. And if the uh, when the water is not turbid and we are suspecting that the count is low, then the membrane filtration method is used, or the volume of water to be tested is very large, then we can also use this method. So what we are doing, we are using a cellulose membrane which is formed, which has different pores of zero point 0.2 to 0.45 micrometers. So when we filter the water, we will filter the water through that cellulose membrane. So bacteria will retain on the surface of the membrane filter. And then what we will do, we prepare a media. Okay, this is membrane lauryl sulfate broth, which we prepare. Uh, it is basically a liquid medium, but we put it in a petri dish, and we also add a absorbent pad. Okay, we take a petri dish and we put a absorbent pad and we pour this medium. Then on that medium, we basically put this membrane filter. Okay, that cellulose membrane that we have used for filtration, and then we incubate that plate for 48 hours at 37 degree. Then after incubation, we will see that there is a characteristic yellow colored colonies of coliform which will develop, okay, which can be counted directly to obtain the idea is that the one bacteria which is present in the membrane filter will give rise to one colony of the bacteria. So one colony forming unit term is used, same as we used in UTI. You know, UTI में semi-quantitative method में we use this term. So we count those colonies, those yellow colonies, and then since we had, it depends कि हमने कौन सा volume use करा है. We can always calculate it for 100 ml of water. Okay. Then colonies are further processed for the detection of lactose fermentation with the production of acid and gas at 44 degree to check again for thermotolerant E. coli. So this is the membrane filter. Somehow my cursor is not working. Okay, so this is the picture showing membrane filtration method. Here you can see that we have these membrane filter. Okay, so this is the filter, cellulose filter. Here, this in this picture you can see this is the cellulose membrane which is commercially available. We filter the water. Now we take the bacteria will retain there. So we will take this filter and we put this in the culture medium, that lauryl sulfate medium, and then we will incubate it. And here you can see these yellow colonies. The pink colonies are not coliform bacteria. The yellow colony will denote the coliform bacteria. So this is the membrane filtration method. Now the advantages are we can test dialysis water, and we can also test for other hospital associated pathogens like which are generally nil fermenter or non-carbohydrate fermenter. And we can also use it for testing clean water and testing large volume of water, because we just have to filter it, you know. And the disadvantage is it is not suitable for turbid water membrane, because then too many colonies will grow on the membrane, and it will be very difficult for us to count the colonies. So then we will have to dilute it. So when numerous colony grow, then we say that it is too numerous to count. Okay, then we will have to dilute that water again and again. We will have to test it. And also it is expensive than multiple tube methods. So these are the disadvantages of membrane filtration method. Now another is endotoxin detection that we do generally in the hospital setting for the dialysis water, and it can be done with the help of gel clot assay, that is also known as luminous amebicide lysate assay. Actually luminous is a horseshoe crab. Okay, so crab के blood cells होते हैं which are known as amebocytes. So generally क्या होता है कि जो bacteria है, the endotoxin of bacteria, they cause clotting of these blood cells of crab. Okay, so these blood cells of crab are commercially present as lysate. So we do it and know it. This test is known as gel clot assay. So the principle behind that, if the endotoxin is present in the in the water, then and we add this lysate in the water, then the gel clot will form. Okay. Then there are other methods like turbidimetric method and chromogenic techniques that can be used. So permissive level which is given is less than 0.25 EU per ml for the endotoxins. Then there are air surveillance method, which are generally done, again same, investigation of an outbreak for research purpose, after reconstruction or newly constructed buildings, after fogging, or for short-term evaluation of change in infection control practices. Okay, so actually there is a ENT lecture, so I'll be little fast now. Okay, so we can do it by passive method, which is a settle plate method, or active monitoring, or the slit sampler method. Okay, main two methods are used. Third is the air particle counter method, which is a new technology that has been developed. So basically this is a settle plate method. It is also known as 1-1-1 scheme, which means 1 hour duration, 1 meter above the floor, and 1 meter away from the wall. So in this, we are taking media, standard media such as blood agar, and we are keeping it in the OT, okay, for the duration of 1 hour. So 1 meter above the floor and 1 meter away from the floor, and we are incubating it at 37 degree for 24 hours aerobically, and then we see the growth. So we count the colony. So here the growth will be there after 24 hours, and we count the colony forming unit. Okay, so these are the standards that has been given in your books as well, so you can read them. Okay, so these standards are given by certain regulatory authorities, which has decided the maximum acceptable level of these colony forming units that should be present in the on the blood agar. So again, another method is active monitoring by slit sampler method, in which this is an air sampler. It is a device in which we basically apply a blood agar plate, and non-volume of air is passed through this machine. So the air will basically pass from that agar plate. Okay, now that agar plate will be taken out and it will be incubated, and then the colony forming unit in terms of volume of air will be counted. So it is generally useful for OT and bone marrow transplant unit. Now, third is the surface surveillance that we do. It is generally done to determine the reservoir of the potential environment pathogen and the sources of environmental contamination. So again, what we do is we basically take sterile swabs, and we soak in this sterile saline, and then we take samples from the different areas. So only pathogenic organisms which are isolated should be reported, because there might be other organisms which can be present. Okay. Now another another discipline of the applied microbiology is the milk bacteriology, in which we must know what are the different milk-borne pathogen and what are the different tests that should be used to detect these milk-borne pathogens. Okay, so again the list is the infections which is primarily of animals. Tuberculosis, brucellosis, salmonellosis, streptococcal infection, staphylococcal infection, cowpox, anthrax. Okay, then there are certain infection that may be due to contamination of milk by ticks or rats. So it can be tick-borne encephalitis or streptobacillus moniliformis. Then again, the infection which is primarily of humans and which is caused by milk, such as enteric fever, cholera, shigellosis, staphylococcal food poisoning, streptococcal infection, tuberculosis, hepatitis. So all these are milk-borne pathogen. So it is basically important to test the milk, for the bacteria which are present. So bacteriological examination can be done by a viable count, by coliform count, by chemical test, and by the detection of specific pathogens. So viable count is basically the standard plate count that we perform. We serially dilute the milk sample and we inoculate it in a special media, which is a yeast extract milk agar, and we incubate it at 30 to 31 degree for 72 hours, and next day the colonies are counted, and number of colonies will total colony count will be equal to the number of colonies multiplied by the dilution factor. Okay, Then coliform count can be done, in which again we serially dilute the milk and we inoculate the McConkey broth again with the inverted Durham's tube, and we detect the presence of coliform with the help of formation of acid and gas. So this is the picture showing standard plate count. So here you can see that we have serially diluted the milk. Okay, 1:10, 1:100, 1:1000, 1:10,000, likewise. So here you can see that the count is also decreasing because the milk has been diluted. This is too numerous to count. Here we cannot count the colony count, but as we are diluting it, we can count the colonies. So here we can see that if the milk has been diluted, suppose here 1:1 lakh, and we have 4 colonies. So the total colony count will be 4 into 1 lakh. Okay. So this is how we do the standard plate count. Now, chemical test basically consists of methylene blue reduction test, phosphatase test, and turbidity test. Methylene blue reduction test is basically testing the presence of bacteria. So if the bacteria is present, it is based on the principle that the methylene blue dye will be reduced by the bacteria. So if the bacteria is present, then the methylene blue dye will get reduced and there will be decolorization of the dye. Okay, so when we will put the dye in the milk, it will first become blue in color, but then slowly it will start decolorizing. So we will see how fast it is getting decolorized. If it is getting decolorized too early, it means it is contaminated with the bacteria and it is not fit to use. Okay. Now, second is phosphatase test, in which we are basically checking whether the milk has been pasteurized effectively or not. So this phosphatase enzyme, it breaks down the disodium para-nitrophenyl phosphate into a compound para-nitrophenol and produces yellow color. Okay. So if the enzyme is present, yellow color is produced. Okay. So if the pasteurization is effective, enzyme will be absent, color will not be produced. Okay, so raw milk will give yellow color and pasteurized milk will remain white. Okay, then there is a turbidity test which checks the sterilization of milk. So in this, if the milk has been heated up to 100 degree, then the proteins which are there like albumin and casein, they will already been coagulated. So upon addition of ammonium sulfate, they will not be able to produce turbidity. So absence of turbidity means that milk has been heated effectively at 100 degree for 5 minutes. So this is the methylene blue test. Here you can see we added methylene blue. This is the positive control, which is blue in color. So after incubation, here you can see that within half hour, if the milk is getting decolorized, it means methylene blue reduction is taking place. So the milk is of poor quality. So likewise, this is also the grading that has been given. So if it is taking more than 5 hours, then it means it is a very good quality of milk. Similarly, this is a picture showing phosphatase test. Raw milk will be yellow in color and heated milk will be white in color. Then we can also detect specific pathogen like tubercle bacilli, and for brucella also we can uh, inoculate them on serum dextrose agar and we can isolate it. And in animals also we can demonstrate antibodies. This must have been told to you by milk ring test and whey agglutination test. Now, the last slide is food microbiology. The primary aim of the food microbiology is to use testing methods suitable to detect, enumerate, and identify microorganism in a food product. Okay, so sample for this, there are no standard procedure as such. It is generally done whenever there is a food related outbreak. Okay, then we do the testing, or we can also do it whenever there is in the food industry, when the food product is formed or when it is processed, then also we can do it. So generally the sample preparation is done first, in which we will homogenize the sample and we will dilute it. A conventional method is cultivation by using enrichment medium or enriched medium. We can also use selective medium, and then we can count the colony forming unit, and then we can identify and type the microorganism. There are also certain newer techniques such as mass spectrometry or molecular techniques which can be used to detect these foodborne pathogens. So this was all about today's lecture. So quickly we will solve few MCQs. So which of the following organism is an indicator for the recent fecal contamination of water? Yes, fatafat say. C, very good. It is a thermotolerant E. coli. Then next is phosphatase test is used to test the effectiveness of pasteurization of milk, sterilization of milk, presence of bacteria in milk, or both A and B. Yes, phosphatase test. Very good. It is for pasteurization of milk. Komal has answered. Okay. Uh, so next is all of the following statements regarding membrane filtration method are true except large quantity of samples can be tested, useful for testing highly turbid samples, can be used for testing clean water and preferred for testing dialysis water. So इसमें से false statement कौन सी है? Yes, B. Okay, it is a false statement that it cannot be used for highly turbid sample. We generally use multiple tube method. Okay, so today actually I will have to end the lecture because ENT has scheduled their lecture at 5 o'clock. So already we are late. So if any question, uh, ma'am, partially treated means actually this is a very vague statement. Partially treatment means suppose someone has done chlorination but they are doubtful about the effectiveness, or they are doubtful whether they have done it properly or not. Okay, so it is a very vague statement. Generally partially treated or can is used, but we are basically using untreated water. Okay, it comes under the untreated water only. So any water that has been treated properly with the water treatment plant through proper procedure, through proper protocol, that will include it in the treated water. So this is a kind of a vague statement. So any other question I'll take quickly and then I'll end the lecture. So shall I stop? Yes, anyone?